Okay, so for those of you who are watching this on YouTube, I just remember to start the recording. You didn't miss much, just the first questions. Anyway, so we're talking about placebo, which is a sham or fake treatment with no effects on the dependent variable. And the purpose of the placebo is to minimize bias and psychological effects, right?
No effects for the
Sorry, what's that? With no effects on the dependent variable. On the dependent variable. Okay. Sorry, let me just see what's going on here with the. Now, given other example where a placebo is needed and then where it's needed, basically when you involve human subjects, you need it. Like if you are testing a new drug, right? The effectiveness of having a new drug, you're gonna need a placebo. Whereas not need a placebo, right? Not needed would be like milk yield in cows with different diets, right? You don't need to have a placebo for things like animals. Yeah, they're not gonna know. Or like, testing the effects of sunlight on plants, you're not gonna need a placebo for that, right? Placebos are only needed when you have human subjects. Again, just two questions on that, very straightforward. Don't get too bogged down by it.
So you don't need to classify in other
Yep. What is a valid hypothesis? A valid hypothesis, we need to have two conditions, right? Testable. And what's the other one? Yeah, falsifiable. That means potentially wrong, right? And you should practice writing hypothesis. Like, if we're talking about the milk yield with the cow, then you can say cows that are fed with higher protein diet will produce more milk. Right? So it's like a statement, right? The most important thing about hypothesis is that you shouldn't be asking a question. There should be no opinions or superstitious stuff in it. Okay. All right. We haven't done this for a while, but there are two graphs here, and you have to know based on the graphs whether or not the drug is effective in reducing jet lag. So here we have a control group, we have a treatment group, and looks like they both have reduced jet lag symptoms, right? So does that mean the drug worked or not worked? Yeah, not effective, right? Not effective. Because both placebo and treatment perform equally well. So what that means is if the placebo does as good as the experimental drug, then why do we need the drug? Right? We might as well just take the placebo, right? It's not really doing anything. It suggests it is just psychological. Over here, on the other hand, this is effective, right? Effective. Only treatment group. Has reduction in symptoms. Okay. That's all. That's all you have to know from lecture 1. Okay, you don't have to study anything else. If you know the answers to these 4 questions, you're covered for lecture 1. Okay, don't waste time. In fact, like I said at the beginning, right? If you know the answers to all the questions in the package, you are pretty prepared already. Okay. All right, macromolecules. It's good to have like one of these big tables in your notebook to summarize all the information. Helps you organize it as well. Let me just make it a little bit bigger. Okay, elements. What kind of elements do we have for carbohydrates?
Carbon,
Mhm.
carbon.
Yeah.
Hydrogen.
Yeah. Yeah. It's always the trio, right? CH and O is in everything. Monomers for carbohydrates, we generally call them monosaccharide.
Glucose.
Yeah, which include things like glucose,
Glucose.
fructose, and galactose. Okay, these are single sugar that you can connect together to build the longer molecule. Biological function. We have a storage of energy. Yeah. In animals, for example. I will just do the example here as well. Okay, it's easier. For animals though. Glycogen, yeah. Glycogen. In the liver and the muscle, right? We can also use carbohydrates for storage of energy in plants. That would be the starch. Of course, starch is also food sources for us, right? We can eat starch. Structural support. We have cellulose. Yeah. Cellulose for cell wall in plants. And there's the other one called a chitin, which is for exoskeleton, right? Like the exoskeleton of bugs or crustaceans, yeah. The crabs, the lobsters, shrimp. Okay. Know your disaccharide as well, of course. Right. The lactose, maltose, and sucrose. Proteins. Okay. On the quiz, I asked you what are the elements that makes up proteins, right? A lot of you put amino acid. Amino acid is the monomer. Okay. The elements are CHO
CO.
and one more.
N.
Yeah, and then some sulfur, okay? Trace of sulfur. Trace means a little bit. Okay. So CHO, nitrogen, a little bit of sulfur. The monomer for amino acid, for protein is amino acid. Yeah, 20 of them. You do not have to know how to draw them. Okay. Biological function, lots of things. Yeah, sure. Enzyme. And we can have an example of enzyme. Yeah. So one thing at a time. For enzyme, an example of that would be like lactase, right? ASE is usually an enzyme. So lactase breaks down lactose. So that's an enzyme. You were saying hormone, right? They form hormones. What's an example of a hormone? Yeah. Very good. Not all hormones are proteins though, right? And some hormones are exactly steroids. Proteins are also useful for structure. Structures like a muscle, right? Nails, hair. We also need proteins for membrane. Membrane proteins. And membrane proteins have like 6 different functions, right? I think there's a question coming up later for that. But we'll put one here, like transport. Right? If you don't have the review, I have extra, okay? Just help yourself. I give it to you last time already, no? You sure?
Is it the same?
Yeah.
Same.
Yes.
Right.
Yeah. Lipids. Lipids, we have CHO and yeah, P, right? Trace of P. Depending which one you're talking about. Okay. Sorry, what's that? N or S? Oh, S is only for protein. Yeah, there's no sum. P for some. Phosphorus for some. Well, in fact, it's only for like phospholipids, right? So the components depends. Right? So there are glycerol. There are fatty acids. Yeah. What else? Sorry. Steroids. So steroid is an example of lipids, but what makes up steroids are like carbon rings, right? Yeah, functional group. Phosphate, yeah. It really depends on which you're looking at, right? So on quiz 1, there was a question where I show you a picture and I asked you what it was, right? Okay, so you have to know what the components are. I'm not going to ask you to do that again on the test, okay? But it probably will show up in the multiple choice. I might, yeah, I might.
The second one on the first one, like, you will provide full limit and ask what is the monomer?
I could ask both, yeah. I could give you I could give you the polymer and ask you what the monomer is. I can give you the monomer and ask you what the polymer is. Both is okay. Okay, function of lipids, cell membrane, right? Cell membrane component, which include phospholipid. That's the major one, right? And a little bit of what? What other lipid is in your cell membrane other than phospholipids? Which other lipid is in the membrane besides phospholipids? Cholesterol. Not a lot, just some. Cholesterol will help change the fluidity. That's right. More cholesterol means more liquid. Do you want to write the quiz now? Are you ready? No? Okay. Yeah, functional group here.
The first one,
Mm-hmm.
I saw the trace of S, I can't remember what you said there,
Little bit of S, little bit of sulfur, yeah.
so. But the comment.
For some, yeah.
Something,
For some lipids, yeah. They not all lipids have phosphorus. Only the phospholipid has phosphorus, right?
I saw. Major, and I saw the source.
Yeah, so in the cell membrane, most of it, majority of it is phospholipids, but some cholesterol as well. Yeah. I need another function, guys, for lipids. Yeah. Storage of energy, absorbing what? Sorry. Not quite. I don't think so. Hormones, yes. Can I have an example of a steroid hormone, guys? Sure. Yeah. Okay. Water repelling surface, right?
That's all.
Some.
But that's all. Okay.
Am I going too fast, guys? Yes. We are being recorded. I'll post it, yes.
We clicked on the link yesterday for the link, but there was nothing.
I posted a link for review yesterday.
The day before yesterday, I think that review report coming to us, but there wasn't a key.
There wasn't a video.
I don't know, but here was a link that you made, there was not. On the news section.
This one, right?
This one, yes.
Here? That's one review?
Let's, here was nothing.
There is no one. Ah,
Now it is a very full package.
No, it wasn't. I didn't change anything. Huh? Some,
it's not available, yeah.
is it still unavailable now? Even now it's unavailable? Oh, sorry, I forgot to make it available. Oh, then you guys email me. Why wait until now? That's okay. I'm on my phone all the time. Some of you, one of you email me on like Sunday, Saturday, 5 o'clock. I still reply. Don't worry. Okay. Yeah, but it's there now. But you didn't miss anything, guys. Okay. It's just the same as this one. Okay. No worries. Okay. Thank you. It's okay. You can always email me up to Friday. Okay. Afterwards, then you will get the out of office reply. Nucleic acid. Okay. What do we have for nucleic acid? CHO always, right?
Yes, need nitrogen, phosphorus.
Yeah. Everything. Okay. Everything in the nucleic acid. Nucleotides, which by the way, nucleotides has the sugar, phosphate, and nitrogen base.
Following that formulas that specify sections that we learn for the base.
There's no,
Sugar, phosphate, and nitrogen base, they have formulas and sections on them.
you don't have to memorize the formulas. No. Instruction make the proteins, that's DNA, right? And yeah, and RNA would be like copy, right? A copy of DNA. All right.
encode
Sorry?
for genetic information.
Sure, yeah. Codes for genetic info. Okay, there are no tricky question on the test, right? It's just basic things. But I understand it's still hard because it's a lot of stuff to memorize, right? Huh? You don't like multiple choice? No, nobody does. Yeah, it's okay. Yeah. Did I already tell you what's on? I did, right? Yeah, 30 multiple choice plus 6 written response question. Okay. Okay, identify the following monomer. What's this one? Yeah.
Nucleotides.
This is nucleotide. Yes. What gave it away? The phosphate, right? Phosphate, sugar, nitrogen base. Okay, I'll write down for you. Sugar, phosphate. You don't have to draw them, okay? I stress again, you do not have to draw them. This one? Yeah, some kind of monosaccharide, right? Yeah, I mean, like, but then, like, how do you know it's galactose and not glucose, right? Yeah, yeah. You don't have to know specifically which one, okay? But like, if you see a hexagon on its own, and you have to, make a guess, monosaccharide would be a good bet, okay? How about this guy? Yeah, steroid. Yeah. Like, I mean, I don't know if it's cholesterol specifically, but I see the 4 carbon rings. I'm gonna say steroid, right? Yeah, and functional groups, right? So there's some functional groups here. That's right. Compare similarity and differences between saturated and unsaturated fat. Okay, make sure you know this one. I mean, make sure you know all of them, but make sure that you know this one. Okay, so let's write it down. I think I'm gonna need more space. There we go.
The one you just passed, are we going to do the labeling or you just write the names down?
What's that?
The amino acid, the nucleotide and all that.
Yeah, what about them?
Are we going to do the label the other one like sugar and all that? Are we doing that in the test like that? I will just name it. Are we going to indicate it? What they are, the sugar, the other one?
I cannot tell you what the question is going to be like, but then I can tell you that you have to recognize them. Yeah. Okay. Saturated and unsaturated. Let's start with, I don't know. Let's start with components. Components, I would say 1 glycerol.
3.
Yeah, plus 3 saturated. Yeah, fatty acids. Unsaturated, we have 1 glycerol plus at least 1 unsaturated. Fatty acids. Okay, you can say glycerol plus 3 fatty acids, but at least 1 is unsaturated. Okay. Do you understand the difference? Right. For saturated, it's all saturated, all 3 of them. For unsaturated, it's also 3 fatty acids, but at least 1 of them must be unsaturated. Okay. You can have 2 unsaturated, you can have 3 unsaturated. The more you have, the more liquid it will be at room temperature. Right? Saturated. Do they have double bonds or no double bonds? No double bonds. Max number of hydrogen atoms. No kinks, right? No kinks, no bends. All straight. One or more double bond. Not max number of hydrogen atoms. They still have room to buy more, right? Kinks in fatty acid. What's that?
Oh, there are no kids.
No kinks.
You know,
What does kink means? Kink like a bend. A bend. Like if you think of the fatty acid tail like this, and then this one, that's a kink. Okay, you can say bend. Sharp bends, sharp kinks. What else? Stay, right? Stay at room temperature. Yep, solid for saturated. Liquid. Example for saturated? Butter sounds good. Sorry, what'd you say? Okay, butter, yeah. And then the other one?
Oil.
What kind? Sure, canola.
Olive oil.
Sure, yeah. Pick your favorite oil, okay? Just not coconut though, okay? Coconut is saturated.
Coconut saturated.
Yeah, coconut is saturated. Okay, but other oil is mostly unsaturated. Okay, canola, olive, grapeseed, avocado, peanut, whatever you like to cook with. I mean, it's never truly fully unsaturated. There's probably mixed with some saturated in it, but like majority. All right, moving on. Again, it's okay if you can't copy everything down. I will post the note online and I'll post the recording as well. If you supply radioactive nitrogen atoms, something that can be traced and detected to a cell and let it grow, what molecules would you expect to find radioactive labels in? Sounds really complicated, but what they are really asking is what macromolecule contains nitrogen? That's basically what this is asking, right? What molecules contain nitrogen? So of the four things, yeah, so nucleic acid, right? You will find it in the DNA, probably some RNA as well, right? Where else are you going to find proteins? That's right. That's it. That's all. Sure. The question is basically asking if you label the nitrogen, if you have a way to see where the nitrogens are, where would they be in a cell? So that you would see them in the DNA, RNA, because those are the things that have the nitrogen, right? You will also see them in proteins. That's all. Regarding DNA, what is the monomer of DNA? Yeah, nucleotide, but specifically, it's deoxyribose. Many people, I would say like about 30% of the class on the last quiz, instead of writing deoxyribose, they wrote deoxyribosome. Okay, that's not correct. Okay, ribose, OSE is the sugar. Okay, deoxyribose plus, yeah, and nitrogen base. ACGT, right? How are nitrogenous base classified? Very good. Purine and pyrimidine. AG. Yeah, you should know this by now. Yeah, CT. And U as well, but because we are talking about DNA, that's why I'm just using T and not U. Base pairing rule for DNA. A pairs with, yep, spells at, right? That's how you can remember it. G pairs with C. What force holds the nitrogen bases together along the length of the DNA? Yep, hydrogen bond. For between A and T, there are two, right? Two bonds. C and G, 3 bonds. Okay. It's something that will be important later on in the second half of the course, okay, when you learn about DNA replication and biotechnology. What is the significance of base pairing rule? The 2 strands are complementary. What does that mean? It means if you know one side, you can find the other side, right? We did this on the test, didn't we? I think so. So something like this, then you can write the other half. TGACCT. The 2 strands are complementary. The 2 strands of DNA.
So are we going to list?
1 second. Yeah, go ahead, Demosi.
Inside the eight.
Oh, I am just telling you that between A and T, there are 2 hydrogen bonds. And this one has 3 hydrogen bonds. So what that means is if you have DNA that have a lot of C's and G's in it, it's harder to pull apart because they're stick together stronger. Yeah. Yes, Doris? You have a...
I was like, you say, what is the significance of the base pairing rule?
significance?
Can we just say the 2 strands are complementary, or we should also add this?
No, I'm just telling you.
Okay, just explain
Yeah,
it.
I'm just explaining what complementary is. Like, guys, don't memorize my answers, okay? These are just like, you know, condensed stuff, right? Okay, use your own words wherever possible. Right? Sometimes I ask students to, like, I used to write out full answers for them. I do that for the study guide, right? Okay, and some people just like memorize that whole thing. If there's like a spelling mistake or a grammar error, they would put that on the test as well. Try to internalize it for yourself, okay? It's much easier that way. Hey, the difference between denaturation.
From complementary.
Mhm.
It's another one again that they are.
Anti-parallel, yeah, but that's not because of the base pairing rule, right?
Okay.
Yeah, that's just a property, right?
That's-
What is the difference between denaturation and dehydration synthesis? Some people are confused about that. Denaturation is basically when you unfold a protein, right? And you can do that by changing pH, temperature, the salt concentration, so on and so forth, right? Dehydration synthesis is the opposite of hydrolysis. Okay. Joints connects monomer to form polymers. Sorry, what's that? Concentration.
Also the, dietitian.
No. You are thinking about factors that affects the rate of, that's different. Yeah, that's like size, temperature, concentration gradient, right? And charge. Yeah. Okay. Many of you are late, okay, but please be on time on Wednesday, okay? You're gonna need the time to complete the test. Okay, be on time, please. I'm so glad there's no TTC strike. I was so worried because I don't know what I would do with the test. No, but I know a lot of students do, right? So it would be a nightmare. That's it, guys, for lecture 2. Okay, that's everything. No need to memorize the 4 levels. Okay, I'm not gonna ask you about enzyme. I already asked you to compare DNA and RNA on the quiz, so I won't ask you to compare again. And yeah, no need to draw anything. 2 down, 3 to go. Okay, list of organelles you need to know. Yeah, let's maybe we come back to this one, okay? I think it's pretty straightforward. It's in your notes anyway. I think I'll do some of the harder questions first, and if we have time, we'll come back. Like, is there any particular one that you don't remember off the top of your head and you want me to talk about? Be on time on Wednesday, okay? For the test. All of these. Yeah, all of this. See, I think some of it is not on here, right? Like the peroxisome is not here, right? So you don't. I know, but it's not me, right? Wait until, next semester, you guys it would be worse. Yeah.
Yeah, just we need to list like just one structure and functions. Like one or two.
Okay, I'll tell you the question, okay? Just because I like you guys, I say that to everybody. No. The question is just matching, okay? I give you the function, I give you the organelles, you just have to match it up, okay? Yeah. Yeah, sure, why not? Yeah. Okay. Yeah. Easy.
Yeah.
Okay. Is there one that you don't know that you want me to talk about?
Yeah.
No? Yay. Okay, moving on. What? No drawings, okay? That's right. Don't draw me a mitochondria, okay? All right, here we go. Describe the central dogma, central theme of biology. DNA to RNA, and then RNA to protein, right? Mm-hmm. This is in the nucleus, right? And this is in the cytoplasm. There you go. Hmm.
That's all we have to show. So
Sure.
that's like say like the DNA gets copied in like the nucleus that gets passed through the nucleus.
I mean, it depends. Look, right? Like on the quiz, it's okay. Like I let you guys pass on the quiz, even though like in the short answer question, some of you just put like, for example, like if I asked some of you on the quiz, what are two functions of lysosome, right? Okay, some of you like really gave me the bare minimum. Okay, apoptosis, digest food. I was like, you know what? Okay, I guess that's fine. All right. But that kind of answer, like, sure, right? But it's not adequate as you move on, right? Like, you're not just trying to pass my course, right? Okay, you're trying to, pass the program and hopefully do well in your next program. So this is really a chance, a safe environment for you to practice those skills, right? Okay. So, don't ask me if this is adequate. If the question is like 2 marks or 3 marks, right? Like, try to elaborate as best as you can. Okay. But like I said, I'm not going to penalize you too much if you just, give me that maybe. Okay. But, again, you're not just trying to do well in this course. Keep that in mind. Describe the structure of mitochondrion and state the function of each part. Okay. Now, we know a lot more about the mitochondria now than we did in lecture 3. And, like I said, I'm not going to ask you to draw it, but you should know, for example, what is this part here again? Not the inside, but yeah, that's right. So you should know cristae contain ETC, right? Right. That's where the purple purple things that we drew on last class. Remember the ETC? Oh, here you can get one. Oh, okay. Right. So the cristae contains the ETC, which is where you're going to be producing ATP, right? Okay. Now, more importantly, we also have the intermembrane space. And the matrix. Now, the matrix is where pyruvate oxidation and what other process occur? Krebs cycle. Yeah. So pyruvate oxidation and Krebs cycle is all about further breaking down glucose, right? And releasing high energy electrons. We're going to capture those electrons to make ATP. Krebs cycle, yeah. You know the third stage, right? The one with the spin. Intermembrane space allows the build up of H+. Yeah. The matrix is where the pyruvate oxidation and Krebs cycle occurs, step 2 and step 3. Cristae contains the ETC, which is stage what? 4. And then we can pump the H+, be on time on Wednesday. palm the H plus from the matrix to the intermembrane space, right? So, in summary, right? In one sentence, what does the mitochondria do? It makes energy, right? ATP.
What is big levels?
Built up of H plus,
Big levels.
which we learned last week, right? We palm all the H plus there, and they really want to come back in, right?
And there was a barrier.
Yeah, there's a barrier. That's right. Yeah, you said that.
What happened?
What happened
How
to
to come down?
we make ATP, right? We make ATP.
Inside.
The inner membrane makes up the crystal. Oh, intermembrane space. Yeah.
Look at the crystal.
1 second. Yeah, go ahead, Demosia.
Are we adding from the matrix to the intermembrane?
Yes,
And then they want to come down back.
that's right.
Oh, they come. Okay.
The ATPase, remember?
It-
The turbine. Yeah. Someone else has yes. You, and you. Yeah.
Question, like, yeah, you said there's another structure, so like, of some of the crystal,
Uh-huh.
so we need to like explain.
If you want to describe the crystal, you would say the inner membrane folds up into these finger-like projections called cristae. Yeah.
It's also.
And then it maximizes the area. Right?
It's also.
So this is the inner membrane. Okay. Blessing, you had a question? Or not anymore? No?
I don't say how did it, I was just like.
Explain it again, before the crystal.
The cristae is just the folding, it's just the inner membrane folded out. Yes. Okay, that's all for lecture 3. There will be no questions on centriole, peroxisome, cytoskeleton. There will also be no labeling. Okay, I'm not going to give you a diagram and ask you to label again. Yes, Jefferson.
People write that they are.
What's that? Go ahead. Jefferson? Yeah.
Don't have to worry about anything that's not on the.
Yeah, if it's purple, you don't have to know.
because, like,
All right. If it's purple, you definitely don't have to know. But if there is something that is not in the review, and then it's not on the purple, like the cell theory, then I would be mindful of it, but I wouldn't spend so much time studying them. Okay. Again, if you're crunch on time, if you don't know how to study all this information, bare minimum is to know the answers to all these questions. Okay. Right. And that's what we're doing, right? So if you really don't have time, just print out this thing that I'm doing right now and memorize all of it before the test. Okay. You should get a 70 at least. If you memorize all these, if you memorize nothing else, but the things that are written here, you should at least get 70. If you study all these things and be able to, express it in your own words, then you should be in the 90s easily. Okay. All right. It's not hard. I guarantee you, it's not hard. Okay. It's exactly what I said is on this. Okay. Just like quiz 1 and quiz 2 is exactly what I told you it's going to be on, right? What's difficult about the course is not what's on the quiz. What's difficult about the course is spending the time to memorize this stuff so you can put it on the quiz, right? Okay. Biology is very different. I don't mind telling you what's on the test because you still have to memorize it, right? Okay. It's like studying for the citizenship test, okay? Or studying for your G1 test. You just memorize everything, okay? It's okay. You'll be fine. You made it this far. Okay, where am I now? Lecture 4. Circle, higher, lower. Okay, we did this. We actually wrote it on the board. Many of you took a picture. It probably is still in your phone. But let's go through it again. If you want to increase membrane fluidity, do you want more or less cholesterol? That's right. More cholesterol, higher cholesterol. And in terms of the temperature, do you want to crank it up or lower it? Higher temperature. Unsaturated fatty acid. Which one is more liquid, guys? Yeah. Unsaturated is more liquid, right? It's liquid at room temperature, right? So the more unsaturated you have, the more liquid it's going to be. And, you know, you want less of the saturated fatty acid.
Sorry. This higher and lower, this last year, I don't understand.
Do you understand unsaturated is liquid at room temperature?
Yeah, of course I know.
So the more unsaturated you have, the more liquid it will be.
She didn't understand that one. But
Lower?
why is now?
Okay, so saturated means solid, right? I don't want it to be solid. I want it to be fluid, right? So that's why we don't want a lot of it. The more saturated we have, the more solid it becomes, right? Okay. Yeah. And then it's opposite for the other one, right? Okay. List 6 functions of membrane proteins. Okay, just list. You don't have to explain, okay? Someone's gonna ask me, do we have to explain? List. Exactly, okay? I would know at least 4 of them to be safe, okay? 4 of them. Okay. So I'll just recite it for you, okay? Attachment, you don't have to write down, it's in your notes, okay? But I'm just like verbally refresh for you, right? Attachment to cytoskeleton. Cell recognition. Cell signaling. Transport. Enzyme. And then cell junction, right? Attachment. Okay. I don't have to write it, it's in the notes, guys. Okay? Yeah. It is in the notes, it's in PowerPoint. Yeah. But no, but it's in the PowerPoint. Okay, lecture 4. Okay, do you guys hear me? You need to know at least 4 of them. Okay, you don't have to explain.
You didn't try? I think you want to plan that.
Oh, do I really have to write it? Okay, I'll write it. Okay, here we go. Cell signaling, right? Yeah. Yeah. Mm-hmm.
I would love if you can just explain. I've read it, because I didn't understand. If there's a way they can just summarize it for us.
Okay.
So I can.
Watch the recording. The recording will have the explanation, okay? Because otherwise, like, it would be too much time. I cannot cover the whole thing, right? Okay? If you forget what these means, go to the lecture 4 recordings. Okay?
Enzymatic.
Yeah, enzymatic activity. Yeah. A bunch of them, right? Close to each other. You guys remember we had that lecture? Yeah?
So this is very, very cool. Nobody is talking about this one. I didn't come to class that day. I tried to read it and
No, I hear you, but here is the recording, okay? Lecture 4, right here.
I've heard it 2 times.
Okay, well,
It's still on before
okay. Ask me a specific question then, okay? Afterwards. Complete the following chart with the following terms. Okay. We had this chart, right? I posted online for you. Not that one. Give me a second. Do you remember this chart, guys? Yeah. So that's a chart again, I'm not gonna repeat it, but you should go through this chart yourself. Okay. I'm not gonna ask you to reproduce the chart on the test, obviously, but you have to know what goes where. Okay. So for passive transport, we have simple diffusion, just from high concentration to low concentration, right? No energy required. Osmosis, facilitated diffusion, and then there are two different kinds, right? Is there one that you don't remember or you don't understand and you want me to explain now? You can let me know.
That's a
Is-
kind of, Professor,
I
kind
don't
of
go over.
is the osmosis.
But like, what do you want me to go through though? Like
Osmosis.
osmosis? Okay, osmosis is basically just movement of water towards a more concentrated end. Okay? So remember this question, right? That we discussed. This is going to be on this particular one. That talks about the water movement. This is basically describing osmosis. If you have a U-shaped tube and the membrane only allows water to pass through, then the water is going to move towards the more concentrated side. Remember I said, think about putting a scoop of salt in your mouth, right? It's going to make you drool. Sucks the water out of your mouth. The water always moves towards the saltier side. Okay, so the water is going to move this way. Water level is going to go up. That water level is going to come down. Okay? And the concentration here, is it going to get diluted or more concentrated? Diluted, because the water is coming over, right? Okay. And this side is going to get more concentrated. Here is what I suggest you to do. When you see a question like this on the test, don't look at the choices yet. Okay. Think about what's going on, write down the key points. Which way will water go? Water is going to go up or down. Which side is going to get diluted? Which side is going to get concentrated? Okay, write it all down first. Then you look at the answer. Okay, otherwise you're going to get confused. So, do we all understand water is going to go this way?
No.
Because it's the saltier end, right?
Okay.
So it's going to keep on coming over until when?
Until the concentration is balanced.
Exactly, until the concentrations are balanced, right? Until the two sides have same concentration. Okay? This is not the right review. Is it this one? Uh-oh, I think I lost the review. Is it this one? Oh, okay, it's this one. Good. All right, so that's it. Oh, there's the question. I went ahead of myself. What are some factors that affects the rate of diffusion? Yeah, we talked about this earlier. There's size.
Temperature.
I charge.
Yeah, I agree. Just four things, right? Again, look at the lecture notes to see how it's being affected. If it's bigger, it's going to be slower, right? If it's harder, it's going to be faster. Okay, explain what happens to this situation. Okay, let's just go through it one more time. Which way is water going to go? Right or left? Yeah, you see the picture is different, right? Don't just memorize what the example is in class. Actually look at it. Which side has more salt?
Left.
The right side
Right.
has more salt, right? So it's gonna go towards the right.
Right.
Right, okay? Yeah, selectively permeable means only water can pass through in this case. Right, so the dots cannot move. If the dots can move, the dots will move to the left, right? But the membrane blocks it. Only water can move. All right, so water moves to the right. A couple other things that will happen. Water level will go up here, right? And water level will go down here. Water level goes up on the right. Water level goes down on the left. Okay.
Point from that water level when just 3 or 4 points, we will write that points.
You write those points?
Like for the answering the question, you will write that points, like, the water level going up in this side, the water level.
Sure, you can say water level on the right goes up, right? Water level on the left goes down. Okay. What about the concentration? Is it going to get diluted or concentrated on the right? Mhm. Okay, and again, it will keep on going when the two sides have balanced concentration. This is the basis for dialysis. People who have to, you know, do blood dialysis because of kidney failure. That's the principle behind it. And that's also the principle if you have like a water filter, you know, at home under your sink, those reverse osmosis stuff. It's basically that. Okay, another one here. Hypertonic, isotonic, hypotonic. Okay. Here, I'll tell you, you're gonna get one of these situations on the test, and you will have to draw them. All right. So bring some colors if you can. Okay. So let's take a look. I will give you one of them. I don't know which one you will get because I don't know which version you will get. Okay. But you will get one of these things. So it's okay. Black and white is fine. Okay. Yeah. 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 Or you can get some from me. Okay. I have pink. Okay. Yeah. 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 But bring your own if you get. Hypertonic. All right. The solute concentration in the solution is blank. The solute concentration inside the cell. If it's hypertonic, what's going to happen? Is it higher or lower? Greater than or lower than? Yeah, this is greater than. Greater than. Okay. We did the whole note, right? With the water tap and everything. Rate of water entering the cell is less than rate of water leaving the cell. effects on animal cell. Okay, so what happens is the animal cell. Okay, let's draw before and after. Give me a second. Give me a second, okay? Oh, I actually have it. I didn't even know. Okay. So here is the animal cell. And then it's more concentrated outside, right? Than inside. Okay, you can put more dots here to make it obvious. Now, ideally, you have two colors. Okay, so you would use one color to show water leaving the cell. And then you'll use one color that shows the water coming in, right? So more water leaves than it comes in, right? So what happens to the cell? Shrinks. So afterwards, the cell shrinks, shrivels up like this. Okay. You can still keep the concentration here like that. This is called plasmolysis. Plasmolysis. Okay. That's what happens when you are dehydrated. When you don't drink enough water, your cell shrinks. Okay. You have cells in the brain that will shrink in response to dehydration. And that makes you feel thirsty. Which one is this again? Is this hypo or hyper? Hyper, okay? Right here, hyper. Shrinks, yeah. What happened? Oh, that's okay. Just don't do that on the test. Okay, isotonic, iso, right? Iso means the concentration is the same. The rate of water entering the cell is equal to rate of water leaving the cell. Okay. Isotonic is the normal state, right? For us anyway. So like this. So if I have two arrows going out, how many arrows should I have coming in? That's right. No change, right? If you were me, would you put this on the test? The isotonic. You know, something to think about, okay?
Yeah.
I'm already very nice. I cannot be more nice than this. I'm telling you. Okay, so no change, right? No change. Now, there was a question. Actually, you know what? I'll come back to it. Let's finish this first. No change. Bless you. Okay, last but not least, bless you again. So hypotonic with a big O, right? This is when the solute concentration in the solution is less than. Yeah. And then this is greater than. Now I need to have a gentle caution for you. Don't memorize less than and greater than, because it depends on how the sentence is structured, right? In this case, solute concentration goes first in the solution, and then inside the cell is second, right? But like if it's the other way around, if I put cell first, right? Like don't memorize it. Try to understand. No, don't laugh. I mean, it's you know, people do that, and I just want to make sure that you are okay with it. Okay. So here is it. It is again. It's more concentrated inside than outside. So like I would have one arrow going out and multiple coming in, right? So then it gets big. Swells, right? And lyse. We can say lysis. It breaks. Right? To show that it breaks, you can you can put some gaps in it. Let me see. There we go. And lyse, L-Y-S-E. Right? Yeah, put some gaps in it so it breaks, right? Okay, there we go. Give me 1 second, okay? I just want to show you something. Is it? Just know the animals, you're okay. Just know the animal cell, you should be fine.
Okay, for the animal cell.
Yeah. Okay, there is a question here. Like these ones, I didn't have time to take it up with you because we were rushing a little bit. But you know, you should go through it yourself. Now here is the one that some people don't understand, and I should explain to you. What's going on? Sorry, give me a second. Okay. So after freezing and thawing some animal cells, what would happen if they are placed in a hypotonic solution, right? What people often do is they see hypotonic, they think of, you know, the stuff we talk about, and they're like, okay, they will swell up and burst, okay? And then they will choose A. But this is wrong because in this particular question, it says after freezing and thawing, right? Okay. So what happens when you freeze things? What happens when you freeze water? Right? You fill up your water bottle all the way to the brim, you put it in the freezer, you know, next day you take it out, what does it look like? Expands, right? Exactly. Water expands, it gets bigger, right? Right? So when you freeze cells, the cells actually expand and the ice crystal from the freezing will puncture, will poke holes in the cell membrane. So after you thaw it, the cell membrane is already destroyed. There are holes everywhere. So it doesn't matter whether you put it in hypotonic or hypertonic or whatever. It doesn't matter. The size is not going to change because it's full of holes. Water will just move in and out, you know, randomly. So that's why there's no change in size. You understand that?
No.
Okay. Which part do you not understand? Give me a question. Huh?
The explanation.
So.
Do you understand when you freeze things, water expands? No? Okay, try it tonight. Fill up a water bottle to the top, put it in the freezer. Next day, take it out. You will see that the bottle got bigger. Okay. So if you do that to a cell, the cell will expand and the ice will poke holes in the cell membrane. Okay, so the cell membrane is full of holes already. It doesn't matter what solution you put it in, the size will not change. Okay, so this is actually not just important for the test, but it's important for your lab. Okay, there's going to be a question in lab that relates to this question. So when you cannot answer things, don't Google it. Okay, just go back to this slide and think about it. Huh? But I just said it's not just because of the freezing and thawing, right? The freezing and thawing will actually damage the cell, right? So it's already full of holes, so it doesn't matter that you put in hypotonic. The water would just leave anyways.
Same thing. Animal cell will not change.
What slide number?
This is slide 37, okay? This is the collection of answers, okay? Slide 37, if you need to see it. Huh? Yeah. Yes, I just posted this morning, yeah. Okay, and again, for those of you who weren't able to see the answers for whatever reason, I posted a new file, okay? It says answer. Because I realize some people don't have the laptop, and if you try to look it at your phone, you won't see the answers, okay? But this one we will. Okay, it's under quiz and test review.
It's locked. One is locked, right?
No, it's not locked. It's fine.
Up,
Yeah.
up one. Number one text you need to do. Yeah.
Yeah, but like I have to post it, right? That's an old one, right? I'm recording right now, so I'll post it after class. Okay. All right. Moving on. Metabolism. Oh, yeah. So no questions on catabolism and anabolism. No questions on IV solutions. Okay, almost there.
Question on metabolism.
No, that's not what I said. I said no questions on catabolism and anabolism. Yeah. Write a balanced chemical equation for cellular respiration. Okay. By now, you should know this. I have trying to, remind you, this is something that you have to know for the test. Okay. How many oxygen, guys? And how many ATPs? No, that's not all. Clearly, which one is reduced and which one is oxidized? What is this? Yes. This is oxidized into carbon dioxide. We saw it in the notes. Okay, you take the carbon dioxide away from the glucose. And ultimately, it's going to go to the water. It's going to be reduced into water. Okay. That's all I'm looking for. Full marks. Okay? If you're able to do this.
That was the keyword. Full marks.
Yep.
Into
Do we have to explain? No, I just want this. Right?
but
Shove right
it's
out.
for-
Yeah. Spoon feeding is great, right? Yeah, you told me write that, okay? For the question. What is the role of NADH and FADH2? These are electron carriers. Here, I'll write down. It's already in the notes anyway, but I'll write down again. NAD plus and FAD captures high energy electrons as glucose is being oxidized. Okay, you should know this by now. What is not here?
Second of the first question.
Oh yeah, sorry. Yeah, the formatting is a bit wrong. Sorry.
What is the role of this and What are the rules? What are the rules?
What are the roles? They capture the electrons, right? And remember the passengers and the taxi, right?
Okay, can we say, send, but that wasn't the question, the question is NADH.
Yeah. So after they capture the electron, they become NADH and FADH2, right? Remember?
Can we use them as, can we use them as the same?
Well, this is like the empty taxi, right? And this is like the filled up taxi, right? Yeah, so there's a little bit of difference.
So when it, you can state it this way, when the answer is like that,
Mm-hmm.
the question forms that way.
Yep.
Okay.
Okay, how about this one? You should know these kind of calculations, right?
Okay.
Mm-hmm. Yeah. Answers, guys? Anyone?
You want the answer?
50 times 3.
NAD times 50 times 3. The other one is 19 times 2.
That's right. 50 times 3 is 150. 19 times 2. 38, right?
188.
Is that okay? Okay, remember, something like this is important, right? Number 10. We did it in class already. Please give it a moment and try it.
Which one?
Next one.
Oh, let's do that one.
Hmm.
With the same, question before, this is the extension of before the, ninth question or not, it is separate.
Yeah, I know. Like it was because it didn't separate, like question 1 and 2 was not separated.
Yes, I know this is ninth question, they show, 50 NADA, N90, FADH. The cycle of this 9 terms or not.
No, this is a separate question. This is a separate question.
So the final answer, ATP was ATP, yes.
Yeah, it's just an S. Remember we did this on the Wednesday, right? And I put like so many stars on that slide, right? You must know it. Okay. This one. 9 turns of Krebs cycle. So let's just go through it one more time. One turn of Krebs cycle, we have 1 ATP, right? This is by SLP, direct production. Yep. Which translates to 9 ATP. This is at ETC, right? 1 FADH2. What do I multiply that by? Yeah. Together, it's 9 plus 1 plus 2 per turn. 9 turns.
We have to give all those explanation before we say what are the ATP.
Yeah, you have to explain it, okay? You have to explain where the 12 came from, okay? Right, but it's good to memorize the 12, right? So like when you work it out and you don't get 12, you know you did it wrong, right?
Okay, is it constant?
Hmm?
You said you have to memorize it, 12, it's very constant.
Is 12 what? I'm sorry?
Is it constant?
Is it constant? Oh, well, yeah, it's constant for one turn, yeah.
Okay, perfect.
Mhm.
Actually, 11.
1 plus 9 plus 2. Right, don't forget to count this one, right?
Okay, the NAD is 183.
I didn't divide, it's a 12 ATP per turn, yeah, per turn. So, you know, not times 9 turns. Hey, bring a calculator if you need to, okay? That's fine. No, it's okay to bring a calculator, okay? But if you forgot to bring the calculator, I cannot let you use your phone though. All right, you have to understand that.
By the-
I didn't divide, it says per turn, here, per turn.
okay, it's fine.
So 12 per turn times 9, right? It's like, you know, $12 per kick, okay? And then you want to buy 9 kicks.
12 times 9.
Then 12 times 9, yeah, exactly. Okay, which step of cellular respiration generate the greatest number of ATP? Stage 4. Which is the ETC. Okay, describe what happens as H plus is pumped from matrix into intermembrane space. I'll type some key points for you, okay? As electrons move down the ETC, its energy is used to pump H plus from matrix into intermembrane space. You don't have to write this down. I'm not going to ask you to write this on the test. You just have to understand the concept, okay? So we know that already, right? Don't we? We use the energy to move H plus from matrix to intermembrane space. This creates high H plus concentration in the intermembrane space, right? As H plus moves back into the matrix through that's basically the 3 main points that you have to remember. This is stage 4. No. Hey, what is the purpose of the final electron acceptor? What is the final electron acceptor? Yeah. It pulls out electrons away from the end of ETC to allow that's all. It's like the story, right? You gotta take the person off the slide at the bottom before the next person can go. That's what oxygen is. Okay. Do you guys have a test this afternoon? No? Can you stay a little longer?
Yes.
Yeah. Okay. If you want to go, you can go. Okay. Doesn't matter. I'm not. I don't care. Okay, that's it for the recording. Signing off now. No, I know, but